Hello, everyone. For those of you still grabbing some snacks, joining us, not pointing fingers, how is everyone doing? Good afternoon. Wow, that's a lot more energy than I was expecting. But I have to say, these couple of days have been a wonderful example of what a beautiful community and culture we have here at the center. As many of you know, we had our external advisory board joining us this week. Hopefully, most of you had an opportunity to engage with them. And today, we culminate with our flagship event, the Geospatial Forum. For those of you that might not know me, I am Georgina Sanchez, Director of Research Engagement here at the wonderful Center for Geospatial Analytics. And it's my distinct pleasure to introduce today's speaker, Dr. Jacelyn Rice-Boayue.
Yes, A+.
I practiced that. And Jacelyn, and I have a cheat sheet right here. Jacelyn is an assistant professor in the Department of Civil Construction and Environmental Engineering. She has served as a Fulbright scholar at the International Institute of Water and Environmental Engineering at Burkina Faso. And she also did her postdoctoral research fellow at Duke's University Center for the Environmental Implications of Nanotechnology. Importantly, she is one of our newer faculty fellows, someone that is already becoming part of our community, engaging with many of our other researchers, faculty, and hopefully soon students. Jacelyn's research focuses on advancing sustainable water resource management by integrating modeling, lab, and field studies with social sciences, but we'll let her to tell us more about that. So please join me and welcome Jacelyn.
Thank you very much. Thank you for the great introduction. I always feel so awkward hearing my bio, but I am here to talk about how I utilize geospatial analytics or leverage them in my research that largely looks to link computational modeling with field analysis and laboratory analysis to understand risk or the occurrence of different emerging contaminants in surface water. And so with that, today's talk is going to focus on watershed-scale surveillance of antibiotic resistance and pesticides. But first, to follow the guidance I got, and I love this question of how has your career trajectory led you to using a geospatial perspective? And so when I first got this question, I went to where did I really first started using GIS for my research, and that was my PhD studies. But then I kind of backed away and thought of really the way that I approach my research is largely shaped by just my personal experience and living in different areas. So we've already kind of discussed my background, but just to note that I have a BS in civil engineering, and then my graduate studies is in civil, sustainable, and environmental engineering, which was at Arizona State University. While I was there, I was part of an NSF-funded center, decision center for a desert city, and it was within the School of Sustainability. And so I was the only engineer as part of a group of students who were from different schools across the university, but largely working in sustainability. And so just how I view engineering problems as kind of interdisciplinary and going across these kind of siloed approaches was largely shaped during that. And I'll even say prior to starting college. So I decided to go to UNLV because my family had moved from Georgia to Nevada when I was about a junior in high school. And it was during that time that I noticed this vast difference in how people thought and spoke about water. So in Georgia, it was really common for people to just drink water out of the tap. I moved to Las Vegas and people really had this notion of don't touch the water. It's not bad, it's not good. Don't drink it. We only drink bottled water. The water was very hard. I would take showers and my skin be really dry, hair really dry. And so that kind of personal experience shaped me into what questions surrounding water quality. And so with that, just to note that We talked about in the introduction that I did a Fulbright at Burkina Faso. I just like to include a map because it is a pretty small country, but it's in West Africa and it's just north of Ghana. And that was also a very pivotal experience that shaped my view of how we look at water quality. So there I was working on research where for certain parts of Burkina Faso, schistosomiasis, which is spread through an absence of sanitation within surface water. So meaning that there are still areas of Burkina Faso where open defecation exists. And so we were looking at kind of this hotspot analysis of where would we expect that to occur, because we had maps of areas, kind of jurisdictions about kind of lack of infrastructure and where open defecation was largely happening. So it shaped my view on how we approach some of these problems. But I will say that my focused entry and my kind of real official launch into building so far my research career within geospatial analytics has been really was launched during my PhD work. And so my PhD work started with this research question of what is the occurrence of de facto wastewater reuse across the United States? So if you're doing anything across the United States, obviously that's a spatial question, right? And so with that, de facto wastewater reuse is the unplanned or incidental occurrence of treated wastewater discharges being in a downstream's drinking water source. And so of course, if we're just localized in a city, you're gonna have your drinking water intake be upstream of your discharges, right? But if you are on a major river and there's a city downstream of you, you may have wastewater discharges that were discharged and make it down to your city. So it's treated wastewater, but we treat wastewater for mainly traditional contaminants centered around nutrients and bacteria, pathogens. However, they're not designed to capture some of these emerging trace organic compounds. And so with that, with needing to answer a research question that was spatially across the US, I developed a model called DRINKS, which was one of those late night PhD aha moments when I got really excited when I figured out this acronym. It gave me the motivation for the last year of my studies. So de facto reuse incidents in our nation's consumable supply. I tried really hard to get it to be a K, but kettle just sounded really silly. So with that, largely this was done with ArcGIS 10. That might date me a little bit, but it was done within ArcGIS 10. Within that, I had data sources for stream flow, come from NHD plus data. And so basically it was set up to work like a stream network, right? So my streams or my rivers and streams, that was kind of like the network links, linking everything, and then geospatially superimposed on that stream network. I had my locations for my wastewater treatment plants. I had my locations for my drinking water treatment plant intakes, and the wastewater was just mapped throughout the system. And so with that, it was supported by a Python code. We did model validation and verification, all those nice steps, but a big piece of this that came out of it was this idea of high frequency of occurrence. So I cut it off to only look at drinking water treatment plants that treated or that served greater than 10,000 people. That left us with about 2,000 intakes. There are several more that serve lower population. But basically, you're able to see that there was high frequency of occurrence, but in general, there was low magnitude, with the vast majority of sites being less than 5%. However, there are these regions where, like the Texas, kind of Rio Grande area, where we do see a lot of reuse occurring. And so from that, there was also this piece, and this kind of ties into me sharing a little bit about my personal background and experience. So I also was just very interested in tying this into public perceptions. So this was a chapter that was driven by my personal interest and a research need, but I had a faculty that is a social science faculty member, and she really helped me with developing a social science survey where we identified 3 cities across the United States. We did it in Philadelphia, Pennsylvania, Atlanta, Georgia, and Phoenix, Arizona. And we created a survey that asked the respondents, are you aware of de facto reuse? We didn't use the term de facto reuse, but we explained this occurrence of treated wastewater and going downstream. We asked, what level of treated wastewater is acceptable? And so the underlying basis here was, or the underlying kind of hypothesis, I should say, was this wanting to know whether people who are aware of de facto reuse are more accepting of planned wastewater reuse. With this notion that there's no new water, right? So if you're aware that all water has been reused in some way, are you more accepting of planned wastewater reuse? And so we did find that just from those 3 cities, the respondents who were aware of de facto reuse occurring within their area were 10 times more likely to be accepting of a planned wastewater reuse scheme. And so with that, this map I like to kind of show as, I'd like to express it kind of as, are these areas of opportunity? Meaning we have a lot of areas where we have the public who are being exposed or drinking water that has de facto reuse. And can we use, some knowledge campaigns to help gain public acceptance for wastewater reuse in these areas? But again, this is mainly the pathway to where I started to look at not only utilizing geospatial analytics for the environmental engineering questions, but also starting to integrate some of the social science perspectives as well. And so with that, to kind of segue into the next framing, what role do geospatial approaches play in what I do? And so this view, so I've kind of started out with my PhD work that linked de facto reuse to downstream drinking water. And a lot of my work now really looks at evaluating those connections between humans and drinking water, or just humans, their choices, how we use water, how we return it to the system, and what that means for either concerns to downstream drinking water treatment plants, so human health concerns, or what it might mean in terms of aquatic health concerns. And so a large part of this work, or framing of this work, is that I like to use computational modeling, geospatial analytics, to inform where in the watershed we should go out in surveillance or monitor. And so we'll do the modeling, we'll identify where in the watershed we should go out and collect samples, collect the samples, analyze, and do more of that like informed, targeted monitoring. And so with that, you'll see that I love this image. I'll go through a little bit about kind of the main research aims that we've been looking at. So the first one, focusing in on the wastewater treatment plant discharge site. So looking at these kind of local aquatic ecosystems here that receive wastewater discharges. So it's a continuous flow of wastewater. And so we compare that to available aquatic health benchmarks for chronic exposures for different emerging contaminants or traditional contaminants coming through a new pathway, which will be the case for the pesticide work I discussed. We've been exploring links between de facto reuse and drinking water quality. So my PhD work was largely centered on the modeling of the occurrence. Since then, I've been trying to answer the question, why is it important, or is it important, right? Also have done some work on stormwater runoff and going more on the mitigation approach of had a project where we were looking at utilizing biochar as a soil amendment to try to capture some of those compounds before they reach surface water. And then under this umbrella of informed targeting field monitoring with modeling. So again, just looking at it through that lens. and also applying an environmental justice lens as well. And so for this talk, I'm going to talk most of the remaining time really focusing on 2 different studies where we utilize these approaches. Starting off with informing targeted field modeling or monitoring through looking at antibiotic resistance and the threat that antibiotics may pose to creating antibiotic resistance in surface water. And so just a couple of slides just to kind of briefly mention what antimicrobial resistance is. So as medicines are used to prevent and treat infectious diseases, these can be antibiotics, antivirals, antifungals. They're in the environment more, or you start to take them more even in your own kind of human environment, and you start to build up resistance. When resistance is built up, the same level of drugs aren't going to have the same impact, right? And so from a human health perspective, that is a real risk or concern. There's been global concern for AMR. It's not necessarily new, but I just have a couple of stories. Bacteria resistant to last resort antibiotic appears in US. That's May of 2016. Drug-resistant superbug may be more widespread than previously known in January 2017. How the pandemic gave power to superbugs, May of 2024. And superbug crisis could get worse, killing over nearly 40 million people by 2050. Not to cause alarm, but more so just to say that, like no harm, but just to say that recognized by the World Health Organization as a threat. So with that being said, there are multiple pathways for antibiotics into the environment. And so for this first part, I'm really focusing on that pathway that leads from the human use to municipal wastewater. Most antibiotics, I think it's estimated about 40% gets excreted, so meaning 40% of the compound or the drug that you're taking actually gets excreted in your waste. And so that would lead it to being in wastewater. And so with that, with the prior work we've done, we have this model where we have estimated across the United States the dilution factor, which is at every wastewater treatment plant discharge site. So for the 14,000 plus wastewater treatment plants, the dilution factor is calculated as the stream's buffering capacity. So the higher the value, the more just stream water you have compared to the effluent. And so with that, we're looking at this because at low concentrations, antibiotics do have long-term and short-term impacts on the environment. And we're mainly concerned about this, the posing of constant selection pressure on bacterial populations. And just to note that this work helps to support some of the ongoing resistance monitoring work. And so what did we do? We took this 3-phased approach, meaning we started off with wastewater treatment plants, looking just at those discharge sites. We then said, okay, well, now that we know what's going on here, how does that look when we overlay that with the other pathways of antibiotics? So we built a Watershed Vulnerability Index to antibiotic resistance. And then last but not least, we took those 1 and 2 or national models. So we took that national model and downscaled it to a model of North Carolina and used it to inform a field study that we completed. And so I'll go through it pretty quickly. This is work that was completed by my prior student, now Dr. Sarah Kamamalek. We looked at 3 antibiotics, ciprofloxacin, erythromycin, and simethicloxazole, SMX. So with those, there's not a standardized or official predicted no effect concentration or bench line for when antibiotic resistance will occur. However, just prior to this study, coming out of Europe, a research team developed surrogates for a PNEC. And so basically we had a benchmark to compare what our estimated antibiotic concentrations are in those localized communities to what we would expect to pose selective pressure towards antibiotic resistance for bacteria. And so with that, we looked at this, and for SMX, the levels coming out of the effluent are low in comparison to the benchmark, so there's no exceedance. However, exceedance is higher, relatively higher for ciprofloxacin. And so roughly 11 of the about 200 HUC4 watershed regions had exceedance above 80%. Here it's color coded, so green is relatively low exceedance, red is higher, and then our dots are showing the erythromycin exceedance, I should say. The comparison here is that this is under average stream flow. However, when we look at a more low in-stream flow, which is, we utilize the low flow statistic of 7Q10, so our 7-day low flow over a return of 10 years, we see that graph kind of light up, right? Because that dilution factor is going to be dependent on that stream's buffering capacity. But that stream's buffering capacity is going to vary temporally based on time of year and drought conditions. And so within this, we see that during those low in-stream flow conditions, the wastewater effluent could pose a threat to antibiotic resistance. And so with that, we then look to combine what we had already looked at for wastewater coming from the population and said, okay, well, what else can we fold into our understanding of the spatial concern for antibiotic resistance? And so with that, we looked at another primary pathway, especially for non-point pollution, manuring, looking at livestock counts. We also incorporated hospitals that were in the area of wastewater treatment plants. And so not to go into it in too many details, but basically we took metrics that were made to kind of aggregate the concern for non-point pollution, so the amount of manure-treated areas, the estimated antibiotic use in this livestock. We had a point pollution index where we're looking at the wastewater flow, the hospital wastewater flows, and the amount of antibiotic prescriptions that are written in that area. You can tell we're doing this at the United States scale, so obviously a GIS problem. Many layers, lots of geospatial data processing and aggregating. We aggregated at the HUC4 scale because we were doing it across the US, which led to, I think, it was a little over 200 sub-regions. We also had information about the hydrologic condition, as well as some estimates for projected climate change. But overall, we utilized an approach where we adapted the US EPA Healthy Watershed Integrated Assessment to make this index. We weighed all of our criteria equally, and basically it's just a relative index. So you're able to see one watershed compared to another, which one is more at risk. At risk here being which one has more of a potential threat of potential or higher pollution, antibiotic pollution within that area, I should say. All right, so kind of briefly, we looked at this, and just a couple things to point out. We can look at non-point pollution. So Iowa, the state, was the highest for non-point pollution, largely associated with a lot of the livestock agriculture out there. And then secondly, for point pollution, we had New Jersey, largely due to the number of wastewater treatment plants that were present. We have more data, I will say. This study was published on environmental pollution, so there's a lot of more detail that we go into about the outcomes of that index. But just for time and kind of to tell the fuller story, so now we had this national model, and we were interested in downscaling that to North Carolina and integrating it into this approach where we're doing a landscape analysis, identifying sites, and then going out in the field, collecting samples at these sites and measuring for antibiotics, as well as antibiotic resistance genes. I had a colleague at UNC Charlotte who did that work. And so with this, we spent a lot of time trying to take advantage of this downscaling approach. So looking into data sets that were available in North Carolina that will allow us to hopefully build up that model fidelity. And so for that, we did this at HUC 10 watersheds instead of HUC 4. And so a couple of quick results. We looked at HUC 10 watersheds in red are more impacted by point pollution. These are, of course, going to be localized to areas where we have our major cities. And then, probably not a surprise to those who have done anything with livestock agriculture in North Carolina, our red is coming up to the top on the eastern portion of the state. And so from this, we looked at our ranking of watersheds, and we aimed to get those watersheds that were at the higher portion of the list, but then also we had to look at feasibility. So we also looked at what watersheds could we reasonably get out and collect water samples from. And so we came up with 3 watersheds that we went out and sampled. And so the first one was Sugar Creek. Sugar Creek served as our watershed that had a higher amount of point source pollution from antibiotics. It has 3 major wastewater treatment plants. This is actually happened to be around the Charlotte area. And then the other 2 that we focused on both feed into the Cape Fear Basin, 6 Runs Creek and headwaters northeast of Cape Fear Basin. They were part of Duplin County and Sampson County, which are first and second for swine producing counties in the United States. So it followed along with what we were getting from our analysis. And what you're seeing is we have sites where we have kind of marked those as being upstream of impacts. So those are the green reference sites. We have impacted sites. Some areas we wanted to have a reference site, but it was going to be impacted by something. It was almost impossible to get a fully free reference, an area like this, where even at our northernmost portions of the streams, we're still seeing CAFOs. But so we have this kind of setup of identifying sites based on impacted or reference. Sarah, not to go into too many details, but we went out, collected grab samples, processed through SPE, and we processed for azithromycin, sulfamethoxyl, trimethoprim, and ciprofloxacin. So with that, we also, again, did do ARGs. I won't present that work, but Dr. Maria Munir at UNC Charlotte did that work for us. And I don't present it because the ARGs were everywhere. I can't make any correlations. The ARGs, they did not follow what we did see with our actual antibiotic pollution. So we learned that we were able to identify within the watershed where we would expect antibiotics to be higher. And so the sites that are, I just kind of drew that box around, those are our reference sites. And so our impacted sites were statistically significantly different than our reference sites. Azithromycin was the predominant antibiotic, accounting for 54% of the total antibiotic concentrations. And I believe it was for azithromycin and maybe SMX. There were 2 compounds where the concentrations that we measured were higher than any of the concentrations that had been reported in North Carolina up to that date. So further just showing that this idea of modeling and trying to identify where in the watershed to take samples and measure these compounds can help us to understand the higher end of those potential aquatic exposures. All right, and so with the remaining time, I'm going to talk about another project. I'm going to go and focus on down the drain pesticides. And so pesticides are not necessarily an emerging contaminant in that pesticides are, of course, applied for many uses. To agriculture, but also they are applied to uses that lend to being a down the drain pathway. And so while the pesticide is not the emerging contaminant, it's this notion that we have a newer pathway, right? So if I were to say, I'm looking at pesticide occurrence, people would think I'm, of course, looking downstream of an agricultural field, right? However, this is the notion that there are several pesticides that are used in our daily life that lend to going down the drain pathway and leading to a municipal wastewater treatment plant. One of those is laundry. So if you've, I guess if you have a worker who is working out in the field, they'll have pesticides on their clothing and they'll wash it. Or I discussed earlier where I went to Burkina Faso while I was there, or before going there, I had to take a whole lot of vaccines. And one thing that I couldn't get a vaccine for was dengue fever, which is spread by mosquitoes. And I was just terrified that I was going to get it. So what did I do? I wore a lot of clothing that was pre-treated for mosquito and insect repellent. So I contributed to this, right? Because that was embedded in the clothing. As you wash it, it comes out and it's going into the wash. If you have a dog, I had a dog for many years. I would always keep her with the flea and tick collar on. When I wash her with the flea and tick treatment or shampoo. When it's warm outside, I'd wash her outside. But when it's wintertime, I'd wash her in the bathtub, right? And that kind of goes down the drain to our wastewater. And then again, on the exposure side, someone who is working with pesticides or even getting exposure from being around their pet, when they use the restroom, it can be excreted as well. And so this work was funded by California Department of Pesticides Regulations, and it was performed by Dr. Denise Ajajonu, who worked with me at UNC Charlotte. And so we're utilizing the same approach of looking directly at wastewater treatment plant effluent location, so that kind of localized aquatic communities. Here, we're applying this through a risk quotient approach where we have an estimated environmental concentration compared to a benchmark. Because this is not a new compound, we do have aquatic health benchmarks for chronic exposures to pesticides that EPA has published. And so based on that, we're able to identify what is the minimum dilution factor that is needed to not pose a threat to that community. And so this is largely based on data that was provided by our partners at CDPR. They have been measuring pesticides in wastewater effluent across California wastewater treatment plant effluents. And so based on their occurrence data, we're able to calculate this dilution factor that's required for each compound. And then the compounds that we chose, bifenthrin, cypromethrin, fipronil, imidacloprid, and permethrin, are ones that were more commonly occurred in their occurrence data. And so with that, I guess I should just say a couple minutes about the motivation from CDPR. So they are in charge of creating regulations for pesticides in California. And so their motivation was, coming from the standpoint of, if we are only making regulations or posing regulations for limits on non-point use we'll say non-point use. So agricultural areas, application that would lend itself to being part of stormwater runoff. If we're doing that, are we missing something by not also capturing wastewater treatment plants? Meaning, if we go out and regulate only the agricultural use, or even the use that is maybe applied to municipal areas on the landscape, or there's pesticides that are applied to buildings. If we're only looking at that and not looking at wastewater, are we solving the issue? So this kind of source allocation or contribution from wastewater, how does that impact, right? Is that also another pathway that we should be looking at? And so to help them kind of answer that question, we calculated that dilution factor required, and then we looked at what the exceedance would be. This time, not looking at a very strong or I don't want to say conservative, but before we looked at 7Q10, which occurs like, I guess in theory every 10 years. However, with this, we just looked at the mean monthly flow that was lowest in that year. So in theory, this is something that happens for roughly a month every year, right? And so this for California, that was stream flow during the month of October. And so these exceedances that we're seeing are based on stream flow during those October flows. And so with that, we saw that of the 165 sites, 115 of those exceeded aquatic health benchmarks without any types of safety factors applied for all 5 compounds. And then we saw that the level of exceedance is going to vary largely based on just the stream size. So Strahler stream order, as I'm sure many of you are aware, is a measure of the size of a stream. It's a hierarchical measurement. So stream order 1 and stream order 1 at the confluence, they will build a stream order 2, right? And so on. And so with that, the higher the number, the more the stream flow, also the less kind of fluctuations in stream flow you would expect. And so it just shows that as you go in these kind of smaller Strahler stream orders, we do have more of a risk of being below that threshold. So this again is like a minimum dilution factor required, meaning if you are under these lines for either of these compounds, you are now it's at risk. So you want to be above. And then just kind of bringing the point home even more with how much that varies over the year, looking at a more wet month of March compared to a kind of lower stream flow condition of October. And then lastly, with that study, what we did was, now that we are aware that we have some areas that are at a higher risk of exceedance for some of these pesticides, California is also a state that has a whole lot of endangered species. If you look at a map of where endangered species occur in the United States, you're going to see it, a lot of it really centralized to California. And so we did some bivariate mapping to compare where we were seeing exceedances as well as how that overlaid with endangered species, and found that 36 of the 41 watersheds with wastewater treatment plants and endangered species had at least one site with bifenthrin levels exceeding the benchmark. And bifenthrin was the one that had the most exceedance overall. All right, and so my last kind of couple minutes, I love this question of what is the future of geospatial analytics in your field? I'm going to give my guess, but I entered this through the lens of a lot of what I presented today is really talking about surface water quality and surface water quality modeling or monitoring. And so the first area that I thought of is this idea of dynamic, predictive, decision-oriented geospatial systems for water quality management. And so there's, a few studies talking about, potential frameworks or architectures for digital twins of river basins. This idea of bringing together monitoring networks, laboratory analysis, sampling, along with computer modeling, remote sensing, UAVs, this Internet of Things, maybe in-stream sensors, and being able to be more predictive, hopefully one day near real time, understanding what the water quality is and being able to have adaptive management based on that. And if that were to happen, geospatial analytics is the root of the basis, the real framework of that. Another area is more broadly, I feel like my work, starting off with my PhD, in essence, was really showing the connection between the different sectors, right? So between wastewater and drinking water, And it goes against this idea that we should be operating in these siloed systems. And so there are kind of these newer approaches or frameworks. One of them is One Water, where we're thinking of this notion that all water is connected. So we should be trying to manage it in that type of way. Another one is One Health. And so linking environmental and health data sets, looking to look at those interconnections between human health, environmental health, and animal health. And these are two kind of big picture holistic approaches. And they're going to take a lot of coordination, cross-sector collaboration, decision support tools and systems. And that is where I hope the future of geospatial analytics is moving, to be able to support this kind of paradigm shift from these siloed approaches to these more interconnected approaches, which better represent the real environment. All right, that is, I think I'm 5 minutes over, but that is it. Thank you so much for having me.
We have a couple of minutes for questions. Raise your hand so that I can bring the microphone. Oh, thank you.
Hi, very nice talk.
Thank
I'm
you.
Ben Clark. I'm actually from your department.
Yeah, you look very familiar.
Right. So I'm curious about what were your sampling methods for the water that you processed for the wastewater project for AMR? And then also, what ARGs did you test for?
Good question. I'll have to get you that list of the ARGs. I can't remember off the top of my head. But for that, we had two different samples that we were collecting. I'll have to look to give you the sample collection for the AMR work. But for the work that my lab did, we collected, I want to say in 1 liter glass bottles. And we were analyzing for antibiotic residues.
Okay.
And so we focus on the chemical compounds.
Thank you.
Hey, I'm curious about the differences between livestock waste and human waste in terms of risk when it comes to like contributing to AMR in the environment. Are they equivalent, or do they have different amounts of antibiotic use?
Oh, that's a good one. I'm not sure. I know relatively what the effluent concentrations are for antibiotics in wastewater, but being able to compare that to the surface runoff from fields, I have to look at that layer. Yeah, when we did it, we didn't compare it from like a concentration basis. We were just looking at it as, where are these areas that have like higher density of the CAFOs? Yeah, but that would be a good thing to look at.
Hi, thank you so much. It was a really great talk. I'm
you.
curious, for any of your project partners, you've worked with a bunch of different folks, when you showed them the results that the water isn't looking great during the October month and that sort of thing, were any changes made, or did they follow up and ask more questions? And just what was the project partnership side of things like?
Yeah, I mean, I'm going to preface this with, it's been a great project partnership in case they're watching. I will say that the challenge, which is a good challenge, is when it comes to publishing. So the paper with the down the drain pesticides, we have kind of reworked how we present the data maybe 3 or 4 times, because it was, in their view, a little too alarming. And I get it. You know, we make some, for lack of having perfect data, we do make some assumptions, right? And so I completely understand. But the good parts of it are that they have, especially a partnership that is there's somewhat like of a municipality, or I say more of a regulatory agency. CDPR in California, they have so much data. So they have pesticides. So within California, it's required for industries to report pesticide use. and it's open data, and they have data also for like monitoring wastewater. So because of that, it's a great partnership in that we had access to data that we wouldn't have had access to, right? And they were very supportive of the work. A lot of this project was showing results, but not only results, but handing over the geodatabase, handing over the data sets. They have utilized kind of our outcomes and building into some of the SWOT models that they're working on. And so it's been really cool to kind of go from you're researching to something might be kind of put in practice, right? And so that's been nice. But I think one of the challenges is just making sure that we're all kind of on the same page as to how things get published if it is showing sensitive data.
Thank you.
Along those lines, I wonder if you've ever explored a shame on you approach, sort of where you're looking at those jurisdictional areas that might be greater contributing to the problem and how that's affecting downstream communities.
Oh, I like that, a shame on you approach. But it's part of, I guess, kind of like a public kind of forward-facing outreach type.
Yes, but,
okay.
and perhaps the idea of identifying where you have watersheds that are greater contributors to this issue, that are, in a sense, bumping the numbers for other watersheds that are not the source of the issue. So in that sense, you're directly affecting your downstream neighbor.
Yes. I have not applied that approach, but I think
Especially
that
if they
is,
are in a different jurisdiction, right?
yes. No, and that is a very, that's a very tough issue too, because then it becomes, if you're in a different jurisdiction, how much power do you have over it, right? So it's going to take a higher organizing, whether that's at the state level or federal level, to really kind of make those connections. Yeah, I haven't, but I'm trying to think if I've worked with anybody who have kind of looked at it from that angle. Not really. I mean, so we're working on a proposal where we're looking at, kind of not really related to this, but identifying, kind of optimizing maybe stormwater BMP placement, right? And so that would kind of be looking at these critical areas. Where are the areas within the watershed that are contributing higher nutrients or whatever the contaminant may be? And for that, it's being done under this kind of underlying assumption that there is some entity that has the power to direct resources to those areas that are contributing more. So if you have an agency that can do that, right, then it's a little bit easier than if it's, a local municipality trying to get somebody upstream. And I'll say, especially with this connection of downstream drinking water, upstream wastewater discharges, you're kind of seeing that come to light with a lot of these emerging contaminants that are occurring at drinking water treatment plants, and there being more of a recognition of this burden that is somewhat placed on downstream drinking water treatment plants based on the uses upstream. But how we balance that, hopefully I'll see it. That's another place for geospatial analytics for the future.
Thanks, this is a great talk.
Thank
You
you.
mentioned you've done some work with flooding, and I was just wondering if you could talk more about the wastewater and contamination as it relates to flooding events.
Oh, where did I mention flooding?
Pretty early in like the first section of your 3 parts.
Maybe it came off of flooding, like when I was talking about like the stormwater runoff, maybe?
Yeah, maybe.
Okay. So I haven't done work with flooding specifically, but this was relating to a project that I had with NCDOT, where we were looking at utilizing biochar as a soil amendment. So this idea that if you add biochar to soil that is on the side of roadways, We can help to capture more of the runoff before it leads to a stream, and also we might be able to treat some of the water as well.
Cool, thanks.
Thank you. Nice talk.
Thank you.
So knowing what you know and all of your background in modeling and local scale and national scale modeling, I'm curious on your opinion. Would you drink Raleigh's tap water?
I do, yes.
Sure.
Yes.
Are there some times of
Absolutely,
year-
especially if they're watching, yes. Yes, I don't,
There's
what
no,
I
like,
do
near-term
love.
health risks to
Yeah,
consuming
no, I will
tap
say, it
water.
is nice to live somewhere that doesn't pop up on my map, so yes, I'm not, I'm not concerned. It hasn't always been the case. Sometimes you're running these models and you're like, oh, interesting, okay. But no, I don't have concerns for the things that I've been discussing.
Do you have a water filter?
Good question.
I mean, most environmental engineers do. But I had it before moving to Raleigh. That's just, the more you know. But yes, I do. See, you said you too. It's just, things you can control. So right now, a temporary one. I used to have one that was installed and then we moved. So right now, we just kind of have a temporary little one that goes on the faucet. Nothing too fancy. Some activated carbon. It's nothing serious.
We have more question over here. Where's the mic?
Hi again. Have you thought about expanding your work into perhaps some other emerging contaminants like PFAS?
Good question. Yeah, in all honesty, I was kind of fighting it a little bit because so many people are working on PFAS. But we have joined. I have a student now working on PFAS and just went out and did some sampling. Yes, so we are really in the early stages, but kind of carrying the theme of today's talk, we're looking at it from a source allocation kind of approach. So the student has been building a model, looking at wastewater, different industrial sites, different land uses within the Cape Fear Basin, and has gone out and done what's called a time of travel sampling approach, where you basically start in the headwaters, and the aim is that you are sampling the same parcel of water as it's going down the stream. And so it's a very heavy couple days of sampling out in the field. And then you're capturing grab samples kind of at the confluence of each kind of tributary along the system. And so you're aiming to identify where these influxes of PFAS or other compounds are occurring. And so, yes, we're very on the early stages of that, but we just did our first round of that approach and they're analyzing samples now.
Exciting.
Yeah.
I've got one. So there was one time that a student and I tried to make a vulnerability index, and we ended up giving up on the actual index step because we just couldn't figure out how to weigh the different factors in a way that we felt was defensible.
Yeah.
And I'm just curious about your experience then in terms of how you combine the metrics to produce the index. Were they just equally weighted, or?
They were equally.
Okay.
I would say the approach that I would suggest is to have like a stakeholder group identify those weights, but within the kind of, bounds of what the study was, we apply to equally weight. We couldn't justify any other. I'm part of another paper that people are working on to, where they're starting out with some of the work we did for antibiotic resistance, but they're kind of enhancing the index. And they're taking another approach where they're starting out with equally weighted, but then they are going through and kind of I want to say randomly, but I'll say systematically, applying more weight to one and less to the other. So like if you have 4, maybe applying like 0.5 to 1 and then splitting the rest of the 0.5 over to 3, and then seeing how that changes your answer. And so almost kind of doing a sensitivity analysis on an index, I guess. But yeah, we took the kind of equally weighted approach because it wasn't a way to really justify doing anything else, I should say.
Perhaps a final question? Okay, hold on. I'm coming to you.
Thank you for your talk. I'm more of a quantitative side of streamflow, and I'm a student in hydrology. And sometimes I heard hydrologic low flow sometimes doesn't mean something for environmental low flow. For example, for some species, the hydrologic low flow is like too low for some species to survive. So it should be defined in a different way. Is there any like research gap or definition gap in between like quantitative low flow or high flow to qualitative?
Yeah, so I'm hearing, so when I'm using the term, I'm more so, I guess, looking at stream flow and how it compares to the historical record, right? But I think what you're getting at is I can be looking at that from a low stream flow of, oh, it's just lower in March, right? But there may be a benchmark as to whether that has an ecological impact for like this baseline kind of sustained ecosystem. Yeah, there was a colleague when I was visiting in Baylor, he was doing some work in regards to trying to set what that ecological flow is needed to sustain. I could give you his name and see if they have some work that may be of interest. I will say, though, the way that has come up in my work is that, so in the very first study where it was actually a chapter in my dissertation where we were comparing the effluent concentrations for endocrine disrupting compounds to benchmarks. And the first go round, we were highlighting, it's a concern here geospatially. And we had some language saying it also serves as a sustained base flow. But so many of the reviewers were like, you have to highlight this kind of juxtaposed position of what low dilution factor means. It means that, yes, you have higher contaminants and could pose a higher risk, but also it means that stream's aquatic life depends on that just source of flow, right? And so what that is, it would be nice to kind of have a benchmark to say, well, at this level, this actually contributes to the local ecology because it helps to have a sustained flow in which these aquatic communities can survive.
And with that, please join me one more time. Thank you.
Thank you.